Okay, perfect.
Perfect. Thanks. Hi. So, I was wondering if you could tell us your name, and your title and what procedures and research you work on.
Yes, so my name is Kim Gokoffski, and I'm an Associate Professor of Ophthalmology at UC Irvine. And I'm a clinician surgeon scientist. So in the clinic, I serve as a neuro-ophthalmologist, which is a subspecialty in ophthalmology. And we treat patients who have a problem with the connection between the eye and the brain. So either the eye can see images, but the brain has a hard time interpreting what it's seeing, or there's something wrong with the brain and it can't control the eye and control eye movements in reaction to the environment. And so my surgical practice includes performing strabismus surgery, which is surgery on the extraocular muscles, which we do to realign the eyes when they don't move in a coordinated fashion. But I also perform emergent optic nerve sheath fenestrations. And so what that is we punch a window into the skin that wraps around the optic nerve to relieve pressure. And pressure can be high in conditions which stem from elevated pressure in the brain. But most of my time, I actually spend running a translational lab whose focus is to develop engineered solutions to regenerate the optic nerve. So the optic nerve is actually selectively damaged in a number of diseases, and because it's central nervous system tissue, it can't regenerate. So when patients have optic nerve damage, they're blind permanently. And so we are developing electric fields into a technology to drive regeneration of these damaged nerves so that they can reconnect with the brain, or to drive integration and regeneration of transplanted retinal neurons to restore vision.
Oh, wow, that's amazing. So, for people who have these optic nerve damage, is it like you typically see people that are young, babies, or older? Like, who do you typically see, and who would your research be directed towards?
Right. So our approach of using electric fields to drive axon growth would actually be applicable to really all types of optic nerve damage. And so the type of disease you're going to see is dependent on the age of the patient. And so we will see pediatric patients who have severe genetic diseases. And so there are a lot of metabolic diseases such as Leber's hereditary optic neuropathy, in which the mitochondria are unable to harness enough energy from our diet. And retinal neurons are the most metabolically demanding cells in the body, so they're going to be the first ones to show a deficit in patients who have genetic diseases. So young patients are unlikely to suffer from things like strokes. And so our older patient population suffers from strokes of the optic nerve or glaucoma, which is also a kind of age-related neurodegenerative disease. And then in the middle, you're going to have the patients who suffer from trauma. So, of course, like young patients who get into accidents, motorcycle accidents who suffer from trauma, or from inflammatory autoimmune diseases. So, oftentimes, women in their 30s will suffer from inflammatory damage to their optic nerve. So, in all of these cases, different sources, but the optic nerve is damaged and patients are permanently blind.
Oh, wow. And so what was like historically done, maybe 20 years ago to treat these conditions? It's obviously this is a new field and a whole new way of looking at being able to treat optic nerve damage.
Right. So I will say that the truth of the matter is that we really have nothing in our arsenal. And so that's why there are millions of patients worldwide who are legally blind from optic nerve damage. A lot of different things have been tried. So, for example, um, with inflammatory causes, we give steroids to stop inflammation. And all of these treatments, and so for diseases like multiple sclerosis, they're pretty effective at stopping the immune system from causing further damage, but none of these approaches are going to restore vision and function to patients because we just have nothing available. There are other groups, especially engineers, biomedical engineers, who are interested in developing BCI, so brain computer interfaces, to try to bypass areas of damage. But as we know, there is it's limited in its ability to actually restore function because of the significant amount of processing and neural processing that occurs at different layers. So, For example, in the eye, the light comes in and is perceived by a photoreceptor cell, which is a specialized type of neuron that can sense photons and changes that into electrical activity. And then that information is relayed through intermediary neurons to the retinal neuron. And those intermediary neurons already process image significantly. And so just bypassing those cells and creating a BCI to interface with retinal neurons leads to really rudimentary recovery of function. So our approach is a little bit different from that in the sense that we're using our devices in order to augment and start up regenerative, natural, endogenous regenerative mechanisms in neurons to try to self-repair.
So it sounds like it's more like a restorative treatment, right? Over so, which BCIs traditionally were more like a replacement treatment, right?
Exactly.
Kind of like making you walk again instead of giving you a wheelchair to return that mobility to you.
Exactly.
So that's super cool. Sorry, I geek out over this stuff, which is cool. So, like, in terms of your research, I guess what would be some of the like? Why don't we have this today? Like, what are some of the challenges that your lab is overcoming, particularly since you are able to see the direct results within the clinic, as well as seeing it even on the microscope, right? Like, so going from benchtop to bedside, what are some of the challenges trying to get there?
I think like the thing that's really exciting about doing work like this is working with people in with different expertise and who speak different scientific languages, right? And so oftentimes we approach problems from different angles and the communication is not necessarily crystal clear between investigators. And so for example, with what we do, so we work on electric fields, and I think that for decades, if not centuries, people have been able to show in tissue culture very reliably that electric fields can guide and direct neurons to grow to where they want to. But it's translating that to in vivo stimulation to be effective in vivo. You can control so many parameters in a tissue culture dish, temperature, pH, right? Charge injection, charge balance, and whatnot. How much charge density, how large your electrode is, right? But there are constraints in a living organism like the brain. You can't put a large electrode in the middle of the brain, right? You want to be very specific about where your electric field is directed, what you're stimulating. And so right now, understanding and building upon the advances of other giants in the field of like which materials we use, what conformable, flexible electrodes we can work with, and like what is that, what I like to call the Venn diagram of overlap of efficacy and safety. That's, I think, a lot of the work right now that we're doing in the lab to translate our work.
Yeah, and so what about like, so I know especially in vivo, things happen that don't happen, like you said, on the petri dish because it's such a controlled environment, like cell migration, right? That's always been a huge challenge, especially when you're sticking electrodes anywhere inside the body. And how do you get them to stay where they want to stay, but over a long period of time, and especially as humans are changing, like we are dynamic systems. So where do you see like your future, the future of this research? Where do you think it can go in 10 years to kind of accommodate for some of these, like, we are dynamic organisms that even at a cellular level migrate, change, right, adapt, etc.
I think that's exactly the question, right? And so traditional approaches for this problem employ genetic strategies to try to upregulate these signaling pathways that were active during development, thinking we can then revert cells into a pro-regenerative state. And these are very cell intrinsic approaches, meaning we're changing something inside a cell. But I equate those approaches to kind of like putting someone behind in a car and putting the foot on the gas pedal and going go, but providing no steering, right? And so there we need to combine and not work in silos and combine our approaches and also apply cell extrinsic. And a major advantage that our engineered approaches provide over these singular genetic approaches is that we can control our treatment not only in space but in time. We can control where we put our electrodes, we can control when we turn them on and when we turn them off. And those things are much harder to do when you take purely genetic approaches, right? But genetic approaches have their advantages too. So the future that I envision is that we're going to take a combinatorial approach, right? Where we use genetics to drive growth and we use our engineered approaches to steer and direct things to where we want them to be. And in addition, a lot of our work is centered towards figuring out how much of what we need to do needs to be implanted surgically versus non-invasively with surface electrodes. You're going to compromise something, right? Efficacy with safety and finding what's the right balance of that. And so the dream is to have a system where we can implant, let's say, a contact lens. Maybe there has to be one electrode that's implanted deeper into the orbit or in the brain. And while patients are sleeping at night, we can initiate our treatment. And so, and also, this is not going to be something that like is one pill, one injection, like fixes everything, right? We can't cure blindness like that. And so the idea is there going to be a closed loop system that is activated while we sleep? And then when we're awake, we can turn it off and whatnot. And so we can, and then will there be also simultaneous things like impedance tomography in order to measure the efficacy of our treatment? So we know how much treatment do we need? When do we not need it anymore? Can we turn it off? And so there's so many exciting directions that I hope to take our research in the future.
Oh, very cool. So just to help us all understand, so efficacy, you mean usefulness, right? Like how useful is it, and how useful is it, like you said, over time, right? This thing of space and time, and how can we play with that to make sure? And then what is impedance tomography? Just
Yeah.
so I know.
So, it's, yeah, no, absolutely. So, like, as tissues heal, their impedance changes. And so, if you can measure impedance over space, over distance, then you can actually potentially record what's the length along, for example, a nerve. Are the axons regenerating, right? And so essentially, can you create a closed loop system where the system can sense how much treatment is needed and then apply that treatment?
So it's kind of like or I understand impedance is like a relative resistance, but at specific frequencies, right? Because the amount of frequency and the amount of input, right, that bounces up and down, meaning frequency, it bounces up and down, changes, like that resistance actually changes depending on that frequency. And so is that how it changes also across space? And then also that input, those frequencies of different inputs, and is that person dependent, or is that like, oh, it's neuron type dependent, or how does that work?
I think that it would be both. And so I think the idea is that you can, when you're measuring impedance over in a certain region, you're getting a snapshot of what's going on in the tissue at that time. And then you can, if you measure it over time, you can see wound healing and whatnot occur in that particular place. But if you're measuring in multiple spots and you know the direction within which a tissue is supposed to heal, for us, obviously, from the eye to the brain, then we can actually track the rate at which our treatments are actually being effective at regrowing nerves. So that's our
Oh,
idea.
cool. That's super cool. Do you see in the future, will we be able to track this? Like you would have to come into the clinic, or would we, like you said, if the closed loop system, you can almost monitor this remotely, which maybe helps people like you're all the way out here in California. Maybe I'm in, I don't know, Wisconsin, and I need the same treatment. Is that how that you would envision that would work, or would you do have to like you have to come in the clinic every month and I have to sit there and I have to measure things and pray that I got it right and I have the right resistances and frequencies and things that I'm imaging?
I think the future is exciting. And of course, being restricted to a small geographic region is not the way that I think things are going. But absolutely, like we obviously, as clinicians and ophthalmologists, we have many tools. We're, I think, one of the best integrated clinical fields with bio and biomedical engineering. And so we like measuring directly. We can see almost our entire organ, right? We just dilate the pupil. And we can measure the actual activity, not only by asking patient responses, but we can measure electrical activity in the retina to the different cell types, to different layers. And so I think that in the beginning, especially during the investigative process, we're going to be very interested in performing those measurements ourselves.
And that kind of gives me a last question, which is I don't know if a lot of people know about your type of position where you're a clinician and a scientist. So you're kind of sitting on both fields of this innovation, but understanding how it actually relates in real life practice, right? I can build the world's best laser, but it's going to be gigantic and it's not going to fit in the room. Or, right, like I can make non invasive sensors that worked fantastic in my controlled environment or just in the subset of patients, the people that I got, to say yes to help me, measure these things. But what's it like being able to see kind of both sides where you're the clinician and you want to make sure it's useful and you know that there's day-to-day challenges, right, of even just being able to see patients versus the innovation side, right, the research side where you can work directly on these solutions, like how can we become a clinician scientist and what does that take?
I appreciate you asking that because I honestly think that like I'm one of the luckiest people here in that I get to do all of this. And so it's really nice to kind of have the scientist and the clinician in one mind because we think almost like we have different paradigms in the way we think, but when you have it in one person, there's just an acceleration. And I think that that's what it is. And it's an acceleration. And it's hard though. It's not to say that it's not. It's hard to collaborate with others. It's hard to work with people who have different teaching styles, research styles, like working hours, right? But at the same time, I love it. I talk to my engineers and they want to, they're excited about a new technology and they're like, Kim, can you, create a thing to this? And I was like, no, that's not going to happen. I can't do that, right? Or they ask me things like, okay, so you put these electrodes behind the eye, is that going to work in humans? And like, of course, like I'm a surgeon. Like I would never have developed a surgical technique that would not be immediately translatable. I can do this in a human, right? And so being able to say that and fast track, that's I think the advantage. Fast track the translation of things is what I think it's valuable to work as teams and be uncomfortable. I think that if you're not uncomfortable, you're probably not innovating enough. And so I think like if I were to say anything, strive to be uncomfortable in your daily life.
That's great. I think that's fantastic. Do you have any final thoughts for our future scientists, physicians, and our engineers who hopefully can help the physicians create these wonderful technologies that you're coming up with, these as all of it, all in one human being?
Well, I mean, I definitely work with a team, so I don't mean to by any means undersell the value and my dependence. I can tell you, I've grown so much with working with the engineers and things that I thought were reasonable, such as like an application of direct current into living tissues in humans is something that I had to be taught, right? And so the learning goes both ways. And I think that the one advice I would give is that life is short, so think big, be bold. Don't bother with incremental. It's not worth it. And go where your passion lies because that's what you're going to do to be the best at.
Thank you. All right.